Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 180th seminar, and I call it a Pompeu Fabra University Day. I am currently in London, have been giving multiple seminars and conference talks and traveling and visiting for 13 days so far, including AICHE annual meeting, Boston University, MIT, Fisk Institute, Harvard University, Cell Press, Imperial College London, Nature Portfolio, University of Warwick, Biochemical Society, SynBio UK meeting, and Queen Mary University of London tomorrow, coming back to United States this Saturday. I met so many great people during this trip, especially many SynBYSS past and future speakers, including Cell and Nature Journal editors, as well as pioneer and rising star speakers. It has been 2 energizing weeks, enabling me to continue it even with 3.5 hours of sleep a day on average, and also multiple journal editors and new pioneer speaker I recruited, and then I guess for the next second conference in Spain, some of the those people will joining, including Nature or Cell journal editors. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Marc Guell. He is a professor at Pompeu Fabra University. He is a true pioneer and expert in multiple topics, especially leading the skin microbiome engineering field. He and I have been collaborating with ONR, US Navy Global Support. More importantly, he is the co-chair and host of the conference I just mentioned, second SynBYSS conference on June 8 to 11 next year in Barcelona, Spain. And as I mentioned, we're gonna have the great conference again with the multiple, journal editors, funders, and of course the big names, speakers, and also most importantly, rising star speakers or young researcher attending that conference. Mark, thank you so much for your scientific contributions and global service and leadership as the conference host next year, and the virtual podium is all yours now. And thank you again.
Fantastic. Well, thanks a lot for this very, very kind introduction. So it's a big pleasure to participate in this legendary SynBYSS series. And yeah, and it's truly an honor to have the opportunity to help in organizing this next conference in Barcelona, which I'm very much looking forward. I think it will be a great event. We have a very nice lineup of speakers, and I'm sure we'll have a lot of fun as well and have very nice science presented. So if you don't mind, I'll present only 3 slides here. I know in the spirit of the series that the important talk is the one that comes after, but I'll just very briefly present a bit what we do in our lab, which, we are very interested in engineering biology. We are especially interested in exploring paradigms to engineer biology, somehow engineer what is not engineerable yet. So we mainly we mainly apply basically these develop tools and apply these tools in 3 main blocks. So one, we have been very interested in the problem of gene writing. So we are very interested in developing technologies to write large messages into genomes, especially combining CRISPR transposons. So in the past we did more DNA transposons. Now we are moving more to RNA, but with the spirit of this large message writing. And as Taisuk also presented before, so we are very, very interested in chassis to engineer the skin microbiome. we have been working with that since maybe 10 years, and here again. So we are very inspired with the natural microbes that we have, and then using them to modulate our basically our skin and hopefully generate interesting functionalities. Also, we do some virus engineering. So somehow, we try to go from molecular principles and then apply these molecular principles both in mammalian systems, but also we really like also to work in bacteria to then deploy them as a chassis for mammalian systems. So I think it's a very special times in synthetic biology. We have had, I would say, actually very exciting last decade. Where, I don't know, we moved from this big capacity of sequencing to big capacity of writing. And this, of course, has allowed to create incredible technologies, right? We went from, so for example, we all know, right? Biology is the best hardware in the world. Nothing in the stars, nothing in other planets so far. We have found that it's so sophisticated that it has this incredible complexity as biology. But somehow this complexity is also difficult to handle. Thanks to DNA, we can abstract this complexity, and then we can transfer modules from organisms to others, right? But so far, nobody could speak DNA, right? We could find DNA in nature, refactor it, and this was extremely successful. CRISPR therapies, right? We will have precise engineering, but at the end, it's the same Cas9 protein that we borrow from a bacteria. Also, incredible things like xenotransplantation, right? We can alter even so genetic properties. But very much in this philosophy, right? Inspiring ourselves in nature and then somehow, basically building tools and refactor these tools. But I think now we have a very special moment with AI, because now, in addition of reading and writing DNA, we can start to speak DNA, right? As humans, we cannot speak DNA, we cannot write poetry in DNA, but now we start to have tools that allow us to write this kind of poetry with DNA, which is, I think, a very exciting point. That's why here in the middle of the slide, I have this synthetic evolution. So I think we can really expand and amplify this Darwinian force, right? Because classically, Darwinian evolution has been on a, let's say, always based on imperfect template-based replication of DNA and then selection. But now with these new tools, we can start to change this paradigm, right? First, with synthesis, the genotype network is completely unconstrained. And with AI, we can write this completely unconstrained genotype network. And we can not only write, but also design, which I think is very exciting. Another very interesting thing is also now when we generate a genotype, normally we need to test the phenotype before knowing how will this phenotype will be. But with methods of learning, maybe we can start to teach even this AI to couple genotype generation to the phenotype, which I think it creates very interesting avenues for synthetic biology. And very quickly, so we have been trying to apply some of these principles. For example, the computer is stuck. Okay, now, so for example, in this problem of gene writing, combining transposons with CRISPR, and then trying to go to nature, as we usually do, right, to find new parts, especially screen them, the ones that are more promising. But very interestingly, recently we could start to then not to stop here, which was the typical stopping time, where we have these better variants, these better parts, but then use this new biological knowledge, put it into a large language model, and start to write, if you will, this new poetry, right? And write better variants, in this case, even better than the naturals, which we are in the very much beginning of all of this, but I am very excited. And then the other system that we are, and then the other thing that I was mentioning, I think coupling this with learning, then not only we have a tool that we can generate, but we can also somehow couple these two very important things, right? Couple the genotype generation to the phenotype by learning, which I think is, again, very much in the beginning, but I think very nice prospects for the next future. And finally, we are very interested in these microbes. This is definitely something we have done for a long time. We especially work with Cutibacterium magnes. We have used to, let's say, alter the microbiome, the composition, and use this as a therapeutic principle to displace certain strains, introduce others, to modulate the skin. Then, again, we are very interested in the tools, in the molecular paradigms. We spend a lot of time in developing techniques to engineer these microbes. And now our focus is more in building modules to do interesting things, try to write these kind of programs that involve sensing. We are very interested in listening to the host, to listening to the environment, and then use these microbes to create effects, right? That could be, physiologic things on the skin, or very exciting project that we are doing with Taisuk, which is more on the thermal domain. So yeah, and I think I'll stop here. So yeah, I'm very much looking forward to Justin's talk.
Fantastic talk, that I really love, your, scope, also breadth and depth, because you are now doing all the hot, topics, including microbiome and also AI, and altogether also the DNA writing. And so that's a fantastic, the research field is kind of broad and at the same time very deep. So thank you so much for your contribution. Also looking forward to meeting you
Pleasure.
again in Spain.
Definitely very much looking forward to meet you.
Fantastic. Great. All right. So now the main speaker of today with a longer introduction. Dr. Justin Panich is a research scientist at Lawrence Berkeley Regional Lab and also served as a deputy director at the Joint Bioenergy Institute, in short, JBA, in Emeryville, California. Originally from Wisconsin, he said, very cold, and he going to the Wisconsin during the Thanksgiving. He grew up immersed in winter sports like hockey and snowboarding. He earned his undergraduate degree in biomedical microbiology and chemistry from the University of Wisconsin-La Crosse in 2013, and Justin continued his academic journey at the University of Utah, where he obtained his PhD in molecular cellular evolutionary biology in 2019 under the mentorship of Dr. Dave Blair. His doctoral research explored the mechanics of the bacterial surface motility. 2020, 2019, he joined LBNL, Lawrence Berkeley National Lab, as a postdoctoral fellow under the supervision of the Dr. Steven Singer, where he focused on C1 metabolism in Necato H16, a key industry organism. Last year, he received an Alpha E Early Career Award through the Ignite program to advance sustainable ammonia production method. This is one of the glorious or the prestigious kind of award for the junior researcher. Currently, he leads basically a research group at LBNL focusing on lignin depolymerization and biological CO2 upgrading to address critical environmental challenges. Also, I believe I met him for the first time in Hawaii, the conference we organized last year, and then he probably also join the Spain conference, probably giving a talk this time. Justin, thanks so much for your time today, and please take it away, all yours now. Thank you.
Thank you for that excellent introduction. Let's see if I can get these slides up. Can you see the slides?
Just give me 1 second. Not popping up. That worked last time, I guess. Can you stop share again and then go to the, yep, coming up.
Okay.
And then slide mode, you can click oh, that's somehow interesting. So whenever you click that one, just disappear. Uh,
Ah,
and then Can you go? Okay.
What is happening?
I don't know what's going on. Let me.
In my case, what I did is I directly shared the desktop, and then it worked. So just sharing the desktop, which maybe is not as convenient.
Try
But
this.
yeah, exactly like this.
Wonderful.
All right.
Working.
Okay, great. All right. Thank you for the invitation to speak today. It's a great honor to have this opportunity. Thanks for the great introduction. Justin Panich, I'm a research scientist at Lawrence Berkeley National Lab. I'm also an MBA candidate at UC Berkeley Haas School of Business, where I'm exploring venture capital and impact investing. And today I'm going to tell you about some of my work mostly that I did when I was a postdoc under Steve Singer on engineering cupriavidis necator and characterizing the CO2 fixation and bicarbonate uptake systems in this host. So first, I just want to advertise my new publication. I'm not going to talk about this in any detail for the rest of the talk, but this is hot off the press, so I just wanted to make an advertisement. There's a co-first author paper that I put together in collaboration with Jay Kiesling's group. We engineered E. coli to take up energy from formate or hydrogen, and we showed that the cells are much more carbon efficient for biomanufacturing when they're able to assimilate this energy, and they also produce less waste carbon dioxide. That was funded through the ARPA-E EcoSynBio program. So here's kind of the roadmap for today. I'm going to give a little bit of background. I could probably speed through most of that considering the excellent introduction that was given. Then I'm going to talk about some recent publications with cupriavidis necator, and I'll talk about some ongoing work with the Bioenergy Technology Office using this organism. And I'll finish up by talking about a really cool collaboration that I am working on with a company called CarbonBridge. So a little bit more about me. I got my undergrad degree at the University of Wisconsin in La Crosse, studying biomedical microbiology. My undergrad research was focused on developing novel antibiotics, which was very rewarding. Then I went to the University of Utah, studied bacterial surface motility, did a lot of snowboarding. In 2019, I joined Steve Singer's group at Lawrence Berkeley National Lab, where I found my passion for working with C1 autotrophs, such as cupriavidis necator. Since then, I've been promoted to research scientist, and I am also the deputy director at the Joint Bioenergy Institute, where I focus on lignin depolymerization and conversion to value-added products. My research group was recently established, and it's focused on combining the disciplines of synthetic biology, chemical engineering, and CO2 electrolysis. Here's a picture of us enjoying boba, which we do pretty regularly. So graduate students, if you come work for me as a postdoc, you get free boba with the deal. So most of my work with strain engineering has been focused on carbon efficiency and dealing with the economic problem that we have with biomanufacturing of commodity chemicals. I have a longstanding relationship with the Advanced Biofuels and Bioproducts Process Development Unit at Lawrence Berkeley National Lab, where I'm able to perform precision fermentation and scale up, and to work with cutting edge gas bioreactors. We've recently been interested in electrochemistry and CO2 electrolysis. So CO2 is a very good feedstock. It's plentiful, it's cheap, but it's inert. There's not any energy in it. If you electrify CO2, you can reduce it and create some molecules that bacteria can more easily upgrade, such as formate or carbon monoxide. And I've been really interested in linking up electrochemical systems to biological upgrading. Most of my current projects are variations on this theme. So got a project that is concerned with making fatty alcohols using a really strange bug that's funded through the Open Collective Foundation, 50 years venture capital. Currently working on a project with the Bioenergy Technology Office, focused on aviation fuel production. I'll be talking about that in some detail today. And on the left here is a picture of me accepting my early career award at the National Academy of Sciences in Washington, D.C. last year, from ARPA-E director at the time, Evelyn Wang. This was one of the proudest moments of my scientific career, and ARPA-E has been an excellent partner for me to ignite my career ever since I started at Berkeley. And I look forward to continued cooperation with them. So CO2 is an ideal feedstock. We have so much of it. It's cheap. It's inert. However, it's cheap and it's inert. And one of the best and most advanced organisms to upgrade CO2 is Cupriavitis necator H16. We have fully developed genetic systems in this host. It can grow on CO2 if you add hydrogen, or on formate, reduced carbon dioxide, which can be produced electrochemically. It rapidly accumulates biomass, and very interestingly, it produces this polyester-like bioplastic called PHB just naturally from CO2, and it can accumulate up to 70% of total biomass in this form, and that's what you see in this electron micrograph down here. Next, I'm going to talk about recent publications, some work from my postdoc. Before I go into the data here, I just want to show you and orient you how we actually grow Cupriavitis necator on hydrogen and CO2. You need a closed system, and if you're doing small-scale experiments in the lab, we do these in these serum vials. We put minimal media, it's basically just water and salt and buffer, into these flasks, and we inoculate our culture. No sugar, no carbon source of any type. We vacuum out all of the air in the headspace, and we put gases back in: hydrogen, CO2, and oxygen. I used to have to do this manually, but now we have an automated platform to do this for us. So it's a little bit less laborious now. And at the end of a 48-hour incubation at 30C, you get these nice dense cultures. So my first paper as a postdoc was a collaboration with Dave Savage's lab at UC Berkeley, the Innovative Genomics Institute. And this was focused on trying to understand how bacterial CO2 concentrating mechanisms have evolved. And an important point to kind of understand that the rest of this portion of the talk is that carbonic anhydrases are essential for most life forms in low CO2 partial pressure. And that's because bicarbonate is a key cofactor for a lot of central metabolic reactions. In cyanobacteria and other types of bacteria that have CCMs, they acquire bicarbonate through these membrane-bound bicarbonate uptake proteins, and there's a couple of different mechanisms for how these work. In Cupriavitis necator, bicarbonate is acquired using carbonic anhydrase-like enzymes. So CO2 can passively pass through the membrane, but bicarbonate cannot. And Cupriavitis necator has 4 of these carbonic anhydrase-like enzymes. So we deleted the main carbonic anhydrase enzyme, and we started to try to figure out how mixing and matching these different parts of the CO2 concentrating mechanism affect growth in Cupriavitis necator. So here's some data. We deleted the wild type carbonic anhydrase, and we don't get growth at lower concentrations of CO2. When you get saturated CO2, the cells start growing because bicarbonate can form just just based on kinetics. Then when we complement back the carbonic anhydrase, we get better growth than wild type. And then when we put in a bicarbonate transporter that was just, characterized by Dave Savage's group from H. neapolitanus, these DAB enzymes that Dave's group has been studying extensively, we get dramatically better growth. So that was a nice finding, and that set me up pretty well for a follow-up paper where we looked in some depth into how the carbonic anhydrase relay system works in Cupravitus necator under autotrophic growth conditions. We developed some advanced genome engineering techniques in this host, and we also demonstrated a completely refactored strain that has different bicarbonate transporters and rubiscos in it. So just to remind you, there's 4 different types of carbonic anhydrases in Cupravitus necator, whose roles are a little bit murky in the literature. So we deleted each of these enzymes individually, and we looked at growth phenotypes at 48-hour endpoints. And we see that 3 of these carbonic anhydrases are mostly dispensable. We see the most deleterious effect when we delete CAN carbonic anhydrase. And when we delete all 4 carbonic anhydrases, we get a similar effect. So then I took my CAN deletion and I just put in a bunch of different bicarbonate transporters and assayed. So these Bacillus anthracis DAB2 and Vibrio cholera DAB2 showed the most activity in vitro in a previous paper. But interestingly, they did not function very well in C. necator. Our best functioning bicarbonate transporters were DAB2 that was characterized in the first PNAS paper, and SBT-A and BIC-A from different cyanobacteria. And these bicarbonate transporters work by different mechanisms than DAB2, which is an interesting finding. And to the best of our knowledge, this was the first time that BIC-A was successfully heterologously expressed in any organism. So then I also developed some advanced genome engineering techniques in CNEC with the help of Yasuo Yashikuni, a PI at the Joint Genome Institute here at LBL. He's developed a technique to put in large constructs into the genome of non-model organisms. We refer to it as CRAGE. And this is how it works. It's a landing pad technology. You conjugate in a transposon that contains antibiotic cassette and Cre recombinase flanked by lock sites, directional lock sites, randomly into the chromosome of the bacteria, and then you can perform sequencing and figure out where it lands. So Cre recombinase is typically expressed on the genome, on the transposon of the landing pad. And the selection to insert your construct of interest is based on swapping antibiotic cassettes. This was difficult in Cupravitus necator. And the problem is that beta proteobacteria tend to rearrange the landing pad. And importantly, they delete the LOXP site, which is crucial for this reaction to work. 6 out of 6 isolates that I had, we had this region deleted, and they were all different deletions. So we went back to the drawing board and we decided to see if we could get in a smaller landing pad that doesn't have Cre recombinase in it. So this is just a kanamycin cassette flanked by lock sites. And 10 out of 10 isolates had that landing pad intact. We expressed Cre on the delivery vector transiently. So this is a vector that does not replicate in Cupravitus necator. It just has your DNA of interest, in our case, DAB2. And I made 2 different constructs. We had constitutive Cre expression and Cre inhibited by lac I. And another problem we had is that our antibiotic selection wasn't really working. We were using a kanamycin resistance cassette. We would get strains that appeared to be kanamycin sensitive but chloramphenicol resistant, but they did not swap out the DNA correctly. So I decided, well, we've got DAB2 and we have strains that are deleted for the carbonic anhydrases. Can we just use ambient air as a selection? Because our carbonic anhydrase strains don't grow in ambient air. And that worked extremely well. So here's our control, no conjugation. And then when we put in DAB2, we get colonies in each of these cases when we express Cre constitutively or repressed by lac I with no induction. And then down here is just a confirmation of phenotypes. So growth on LB as you'd expect. And we would expect no growth on kanamycin. That would indicate that the kanamycin cassette was swapped out with DAB2. We see this in one out of 4 isolates when Cre was constitutive, but we see this phenotype in all 4 isolates when Cre is inhibited by lac I. So you need just a light touch of Cre to make this work in this organism. And here's the PCR gel to confirm. So that enabled us to free up some of the epigenetic space. Now we have DAB2 on the chromosome. And we have this strain that's deleted for both copies of Rubisco, along with 2 carbonic anhydrases. And that allowed us to put in plasmids that express Rubisco. And we were interested in how many different types of Rubiscos can actually function in the strain. So we were surprised to find that C. necator was able to express Rubiscos of different geometries. Alrubrum and Arspheroides Rubisco were the best performers. Gallianella, the Gallianella Rubisco is the fastest Rubisco that has been characterized to date in vitro. It did not work in our organism, which was an interesting finding. So in summary, we demonstrated the flexibility of bicarbonate acquisition and CO2 fixation in this host. We demonstrated a variety of Rubisco geometries work in C. necator. The main finding here, though, is that bicarbonate acquisition seems to be the real limiting factor for autotrophic growth in this host and not CO2 fixation. So next, I'm going to be talking about some current work that is ongoing with the Bioenergy Technology Office using C. necator. So I'm a part of the CO2 reduction and upgrading e-fuels consortium with the Bioenergy Technology Office of the Department of Energy. This is a large consortium featuring several different national laboratories. And there's 6 or 7 different projects that are ongoing in the consortium currently, ranging from CO2 electrolysis to biological upgrading, aerobic and anaerobic upgrading. So there's a group at NREL that's focusing on anaerobic upgrading and feeding of carbon monoxide. So generally speaking, we're interested in using CO2 runoff from corn ethanol production and turning it into carbon monoxide or formic acid and performing biological upgrading. So as mentioned, there's a group at NREL that's focusing on anaerobic upgrading of carbon monoxide. We're focused on aerobic upgrading using formic acid and Cupriavidis necator. So we primarily collaborate with Chris Johnson's group at the National Renewable Energy Laboratory. There's also Adam Gus's group at Oak Ridge is also involved. Our local group at Berkeley Lab consists of Eric Sundstrom, who is the PI, and he is a staff scientist down at the ABPDU. I function as a technical lead on this project, and Bassem is a very gifted postdoc who's a process engineer who performed most of the experiments that I'll be showing in the next few slides here. So Down at ABPDU, we have HEL gas bioreactors. And these are, we have 8 different towers. They have capacity up to 500 mils in each of these towers. You can co-feed up to 4 gases at a time. Operation up to 3 bar. It's fully automated except for taking optical density measurements, just because we couldn't fit much more into these towers here. We have integrated micro GC, so we can get off-gas analysis in real time. And what you're not seeing in this picture is behind this wall, we have several different gas tanks, and this is a layer of safety for the researchers. So if anything were to happen with, say, hydrogen, there's a brick wall between us and the hydrogen. We also work with these carbon monoxide detectors if we're working with carbon monoxide. And the problem we're interested in here is making formic acid growth more carbon efficient. It's a little bit counterintuitive, but the stoichiometry of bacterial metabolism in this case is not great. So if you oxidize formic acid, you get 1 CO2 and 1 NADH, but you actually need quite a lot more energy than that to fix carbon dioxide. So right out of the box, if you're putting formic acid into a bioreactor with CNEC, you'll get about 60% of the carbon as CO2 runoff, and about 40% of the carbon in biomass. So that's a real problem if your main objective is to convert carbon dioxide into a value-added product. So we're approaching this problem by co-feeding hydrogen and formic acid, and that will provide the cells with additional energy equivalents to fix the carbon dioxide into biomass. This is what our setup looks like. So we inoculate the cells in here with an initial concentration of formic acid, about 20 millimolar. And then we feed in 35% formic acid and 250 millimolar ammonia to keep the pH level. We have pH control here, and we're also co-feeding hydrogen gas and oxygen at half of VVM. And then we're also able to analyze the off gas using our micro GC. So we've got some promising initial results here. So when we grow on just formic acid, we get about 10 grams per liter biomass. We get about a 30% bump when we co-feed in hydrogen. And then when we pressurize it, we get an even better result. And we're able to show biomass yield increases of about 50%. We're able to do some mass balance here, and we can analyze how much carbon goes to biomass and how much carbon goes to CO2. So with formic acid alone, we see about 60% to biomass and about 40% to CO2. When you add hydrogen, that ratio flips. So more of the carbon is going into biomass than CO2 runoff. And when you pressurize it, it works even better. We can analyze CO2 production. We do show a decrease in carbon dioxide evolution off of the stack, which is exactly what we would expect. And we see a similar result when we normalize to moles of formate input. So in summary, we've dramatically shifted the ratio of carbon that we can put into biomass and therefore value-added products by a significant factor. Next steps with this work, we're going to be collaborating with Chris Johnson's group, who've developed some strains that can produce precursors for aviation fuel. So just to wrap up, I want to talk about a collaboration that is just starting, and I'm really excited about where this is going. So I've been working closely with CarbonBridge. And I just want to point out, I do not have any financial interest in this company. I just think they're doing very cool work, and they're thinking very hard about the bioreactor problem we have in this field. So they've developed these bioreactors, Manu and Sophia, that are direct gas bioreactors, and they have this advantage that they're these compact server-like units, and you can stack them and scale out rather than scaling up. So one of the main problems with commercializing any technologies based on cupriavidis necator is you put it's extremely capital intensive to buy these gas bioreactors and to scale them. They're usually one of a kind items, and investors get very nervous when they dramatically increase the spend with no real risk aversion, because you can build a $100 million bioreactor and find out that it doesn't really work. So this technology is exciting because it tackles the financial problem in this field through a different angle. So bioreactors are ripe for innovation. So bioreactors were developed by basically the beer and cheese companies many decades ago, and there has not been much innovation since. They're expensive, and they tend to break easily. The real problem, though, if you're going to be using gases as a feedstock, is mass transfer. So hydrogen does not go into solution very easily. This is also a problem to some extent with oxygen. But the pharmaceutical companies that use these bioreactors aren't concerned with that because their products are so valuable that they don't care so much about efficiency. So what CarbonBridge has done is they've developed a way to immobilize cells inside of these bioreactors, and you can just simply flow through your gases and then remove your product of interest from the chamber by flooding the chamber. Here's just a kind of a visual representation. So if you eliminate the step to dissolve the gases into liquid, then more of the gas and carbon substrate should reach the bacteria and should become a product. This company is broadly focused on methanol production, and they're able to show a dramatic increase in methane to methanol conversion using a microbe in this host. They're already 400% more productive than the best published data. So they're currently supported by the ARPA-E Green Wells program, but we're trying to get some more funding to adapt their system for cupriavidis necator by creating a kind of a stainless steel version of their unit that can be used for hydrogen and carbon dioxide fermentation. So with that, just give some acknowledgements to mentors that I've had over the years: Steve Singer, Eric Sundstrom, Blake Simmons. It's been a pleasure collaborating with Jay's group, Lab Ops, and other admins at JBay. My group has been excellent to work with as well. Really have enjoyed my time with them. And I'd also like to thank all financial supporters, ARPA-E, JBay, BETO, 50 Years Venture Capital, and Energy and Biosciences Institute funded most of my postdoc work with Dave Savage's group. If you'd like to connect with me on LinkedIn, I'd be happy to continue the conversation. Reach out to me at via email at any time. And yeah, with that, I'll take any questions.
Fantastic talk. Amazing. Let me check. Mark still there. Oh, Mark, please ask question or the comment.
Actually, I had a question. Yeah, maybe it's a bit naive. This is distant from my field, but I was very curious. So when you were talking about this challenge of the mass exchange because of the gas, do the people develop some kind of carriers? Because here I couldn't avoid to think about hemoglobin, right? Somehow humans have solved this by using a protein to somehow, and I was wondering if there are some of these approaches that have been explored for, yeah, helping in this problem.
What do you mean by carrier?
So, for example, if it's difficult to make a liquid gas exchange, could we use some chemical, for example, oxygen to carry it in the blood? We use hemoglobin, right? Could we have such
Yeah,
a thing to carry hydrogen or to carry, I don't know,
yeah, yeah. So that's a really good thought. There's a group at MIT. I'm not going to name them, but they're focused on using these metal organic frameworks to make hydrogen more soluble in bioreactors. I think that's a really promising angle. Um, Other than that, I haven't heard too much on that end. I mean, I guess you could always grow your cells in oil if they can stand high viscosity. That's an approach that might work. But I'm most bullish on this metal organic framework research that's ongoing at MIT.
Oh, very interesting. Can I ask another small question?
Please.
And also, you started the talk discussing these enzymes, also somehow to put like, for example, the carbonic anhydrases. So, uh, what is the let's say the distribution of activities that we see in nature? So, are we seeing like some extreme properties, some that are super efficient? For example, in Cas9, right? We have Cas9 from S. pyogenes that is super active, whereas most of the Cas9s are not. So, do we have such an exception for the world of carbonic anhydrases?
We do. It's a great question. So carbonic anhydrase is actually like the fastest enzyme in nature, just out of the box. The problem is it's reversible in Cupriavitis necator. The DAB and the bicarbonate transporters that I discussed are vectorial. So it those function to, we believe they function by pumping bicarbonate into the cytosol semi-irreversibly. Kind of gives them a boost. Whereas if you're just expressing a carbonic anhydrase, the rate itself doesn't matter so much because it's a reversible enzyme. Yeah, but even human cells have carbonic anhydrases. You can open any biochemistry textbook and it'll be an example for, we need them to keep our blood in the right pH range.
Thank you.
Mark, do you have any other question?
No, that were my two questions.
Okay, so let me ask one question, some practical question. Actually, I'm really glad you mentioned many people I also interact, including Jay, Blake, also the Yasuo. But this question is basically about the Yasuo's technology that is about craze, right? So I recently connect, actually that's this week, we just briefly chat over the online. The question I have, he actually invited me to consider that technology craze. Based on your experience, what kind of thing should we know to make that technology implement successfully? Because you mentioned something, but to the general public, general community people who wanted to use that one, including myself, what insight you got and what kind of thing you need to avoid, etc., to make that more efficiently implemented?
Yeah, it's a good question. From what I have understood with my conversations with Yasuo is it works out of the box in most hosts if you have good selection cassettes. Cupriavitis necator was a huge pain to make it work, and it's primarily because beta proteobacteria tend to rearrange the landing pad. And Yasuo has seen this in other beta proteobacteria. He doesn't see it so much in the more common hosts like the gamma proteos. He's gotten it to work in alpha proteos. So I'd say like just make sure that just make sure that like your host falls within a class. Just if it's not a beta proteobacteria, it'll probably work. I
I
guess
see.
that would be my recommendation.
So the thing is actually he mentioned I'm mostly working on nowadays. Of course I'm working on multiple different bacteria, including Pseudomonas, E. coli, also the some yeast, also the mainly the Rhodococcus. That's a gram-positive one. Unfortunately, his list of the bacteria does not include the Rhodococcus. So he actually even said, I need to do something. Rather than relying on their kind of people. So I just wonder what kind of thing we need to understand to make that happen better. So that's why I'm asking the question.
Yeah. I mean, if you could conjugate it in a transposon,
Hmm.
then my gut feeling is it's going to work fine.
I see. All right. In that case, I need to try that one. My second question is, you kind of talking about the gas, C1 gas, conversion at the end of the day. And then you're also talking about some, the bioreactor configuration briefly. The one big example is the now, LanzaTech doing, fantastic job. Can you kind of compare, the one you just, mentioned briefly and then, LanzaTech's technology? What's difference and what's the similarity?
Yeah, that's a really great question. So LanzaTech, the main difference is they're using anaerobic hosts. They're using Clostridium.
Right.
So that makes it quite easier to work with because you don't have oxygen.
Mm,
You mix hydrogen and oxygen, you know, it could go boom.
mm,
Uh, you need to be a lot more careful with safety procedures if you're using Cupriavitus necator. Most companies that work in this space will work in a non-explosive range, like they'll just use low concentrations of oxygen.
mm,
Um, but even then, like they're operating at about 100 liters.
mm,
I think Novo Nutrients got to be, I think they were at 300 liters or so
mm.
with their aerobic process before they ran into some funding issues. Um, but in general, LanzaTech is able to retrofit old reactors that have been used for ethanol production.
Mhm.
Whereas you don't kind of have that luxury if you're trying to use
Uh,
oxygen. The real advantage of using oxygen, though, is you can more easily make complex molecules. You have a lot more energy around. So if you want to directly make a terpene, that's within the realm of possibility.
mhm.
It's a little bit more difficult with the anaerobes because they use a lower energy pathway, but I've seen there there's some work out of NREL where they're starting to be able to make some more complicated molecules. I don't want to talk too much about it because somebody else will probably come on
Sure,
SynBYSS and talk about it at a later date.
sure.
But yeah, that's the main difference.
I see that clear my my kind of understanding much better. So I kind of a little bit know about the Ranja text word and the technology. And also you mentioned about safety measure. I mean, during your talk. So now that makes sense for now. All right. I guess I mean now I probably need to close because I'm now very hungry. So let me close and then let's just chat afterward. Okay. Thank you all for joining and staying today. We'll meet again on actually November 18. Also, this is a off time again, the Tuesday. But the same time, that means 10 a.m. Chicago time with the same Zoom link. That is because of the government shutdown. We couldn't do that in a seminar, during the shutdown. So we'll have the Dr. Diane Okamuro at National Science Foundation and Professor Lu Kong at Stanford University. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us and promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thank you. And I stop recording. Just give me 1 second.